Welcome to the HNRCA Monday seminar. It's my great pleasure to announce our speaker today, Dr. Roger Fielding, who is indeed a senior scientist in the Metabolism and Basic Biology of Aging Directive here at the HNRCA. Dr. Fielding is the associate director of the Boston Claude Pepper Older Americans Independent Center, in addition to a professor of nutrition in the Friedman School of Nutrition Science and a professor of medicine at Tufts University School of Medicine. Dr. Fielding is internationally renowned for his work in underlying mechanisms contributing to the age-associated decline in skeletal muscle mass. He has been very prolific. He's published more than 300 peer-review papers. He's deputy editor of the Journals of Gerontology, Medical Sciences, and Calcified Tissue International. He has also received multiple awards, including 2 from the International Osteoporosis Foundation, and most recently, he has just been awarded the Excellence in Rehabilitation of Aging Persons Award from the Gerontological Society of America. Today, Dr. Fielding is going to talk about a precision medicine approach to resistance training and its role in prevention and treatment of sarcopenia. Before we start, I just want to ask our participants to put your questions in the Q&A, and we will have a Q&A discussion at the end of this seminar. And with that, I turn it over to you, Roger. We really look forward to your seminar.
Thanks so much, Sarah. And thank you for inviting me to give this seminar today and sort of help me consolidate some of my thoughts around the heterogeneity and the response to resistance exercise training, and talk to you a little bit about the etiology of a new project that we're very excited to be starting in the next couple of months. Just a few disclosures to start. And I thought it would really be appropriate to just start with a little bit of background about why I've been interested in this area for the last 30 years. And this is just an update from the US Census about the prevalence of a condition that we refer to as mobility disability in the United States. And from this 2018 report, which is from 2014 data, census reports are that about 32 million people living in the community had difficulty walking a quarter of a mile. And this is all by self-report, including about 18 million of them who could not perform this activity. Correspondingly, about 29 million adults had difficulty climbing a flight of stairs, and 4% of these individuals were unable to perform this activity. So among individuals particularly over the age of 65, so older Americans living in the community, about 39% of them have some difficulty walking or climbing stairs, of which about 10 million of those people report that they are unable to do these activities. I think it's interesting from a biological perspective that the motor proteins that are responsible for muscle contraction, movement, mobility, locomotion are really, this process is really the result of several very highly conserved proteins across species. So whether you're an insect, a bird, a fish, a dog, or a human, you're really relying on a very highly conserved number of skeletal muscle proteins that are responsible for contraction and for movement, locomotion, and mobility. And interestingly, across multiple species, there are observed declines in motor function with advancing age. We see this very clearly in the Drosophila, which has a very short median lifespan of about 50 days. And you can see that mortality really occurs when their flight index or the frequency of their wing beating frequency declines. If you look at survival time in rodents, this is in rats. This is from Christy Carter, who's now at the NIA. But when she was at the University of Florida, she did some very nice studies looking in rodent models at at a summary physical performance score, looking at how survival time was linked to higher physical performance scores in this summary performance in rats. And the same is true in humans. If you look at something as simple as walking speed or habitual gait speed, you can see that at any given age, there is a survival benefit for faster walking speeds. And this is a large individual subject pooled meta-analysis that was done by Stephanie Studensky. And what's interesting here, this is true in both males and females, and this survival benefit of faster walking speeds actually persists well into old age. This is true in individuals in their 60s and 70s, but it's also true in individuals in their 80s and 90s. So a pretty robust effect. I think it's interesting that sometimes mobility problems fly a little bit under the radar, although with the changing demographics of our older population, I think we both personally and professionally are observing these. I think it was interesting that on or near the failing health of Queen Elizabeth and in the months leading up to her death, it was really interesting to me that the reports from Buckingham Palace were that Queen Elizabeth was unable to perform some of her duties, and this was really a result of her continuing struggles with issues getting around and clearly declines in her mobility and having mobility issues. So this is becoming more timely, and I think there is more awareness that is occurring about mobility problems in older individuals. Now, we'd like to think, and we believe in part, that the changes in skeletal muscle mass and function, or this syndrome that we call sarcopenia, is really the underlying substrate for some of the mobility changes that occur with aging. This is just an illustration, and if you've seen me give talks before, you maybe have seen these slides, but I think it's worth repeating that there are very good visual images of what happens to skeletal muscle as people get older. This is a 43-year-old male with a BMI of about 25, normal body weight, and a 76-year-old male, again with the same identical BMI of 25. And you can see this is a CT section from the mid-thigh. The white area in the middle is the long bone of the leg, the femur, and you can see that the blue, dark blue and light blue is skeletal muscle, the muscles of the anterior aspect of the lower limb, the knee extensor muscles, and posterior aspect of the lower limb, the knee flexor muscles. And you can see quite dramatically that there is a clear reduction in the cross-sectional area, the size of the voluntary muscles of the lower limb, both the knee extensor muscles and the knee flexor muscles. And interestingly, there's also some increased deposition of adipose tissue that occurs around some of these large muscle groups, and some qualitative changes in the actual optical density of these images, suggesting that in addition to the absolute loss in muscle cross-sectional area, there are also some corresponding increases in the intramyocellular fat content that occur with aging. And those have important implications not only for physical functioning and muscle performance, but also for the metabolic engine that skeletal muscle is with advancing age. So I think right now there's generalized agreement that sarcopenia, or the syndrome of aging that we call sarcopenia, is a syndrome that's characterized by progressive generalized loss of skeletal muscle mass and strength, with the risk of adverse outcomes such as physical disability, poor quality of life, and death or mortality. I think after many years of discussion and deliberation, we still don't have an absolute consensus definition of what sarcopenia is, but I think in general, there's pretty universal agreement that the components that we think about when we're trying to assess sarcopenia and evaluate that in individuals are really the loss in muscle mass, the declines in muscle strength, and the declines in physical performance. And there have been numerous novel approaches to this definition that have come out of several different working groups, both nationally and internationally. And I've participated in some of these. I think it's important to remember when you're talking about syndromes, the current consensus definitions are really based on consensus or expert opinion. They've all included objective measures of muscle or lean mass. They've all incorporated muscle weakness or reduced physical functioning into the definition. But until recently, sarcopenia criteria and cut points to define sarcopenia have not necessarily been evaluated with respect to their relationship to clinically meaningful distal outcomes like falls, fractures, and mortality. I was able to participate in some recent work that did this. I just want to share this with you, and it's important for when we talk about some of the data that we've generated and how we've applied these cut points when we talk about markers of cellular senescence and biological aging. And just to show you this has been the evolution over the last 10 or 15 years or so, where many of the reports about definitions of sarcopenia were really a result of expert consensus. I led one of these groups in 2011. There was a group in Europe that did a very similar thing right around the same time. But as time has moved on, there has been more embracing of evidence-based approaches to defining what sarcopenia is and what the relevant cut points and definitions are that should be applied both in research and clinical settings. So I think we've made progress. It's clear that we still need to make more progress in this area, but I think in addition to increasing awareness among patients and clinicians, we've made some progress in identifying and defining what these terms are. The Sarcopenia Definitions and Outcomes Consortium, which I participated in, used an evidence-based approach to define some criteria that predicted poor physical performance and was associated with bad distal clinical outcomes in a very large, multiple cohort, individual patient analysis study. And it's important to note that in that consortium, we did define some objective cut points for sarcopenia around isometric hand grip strength. And in men, this was a grip strength less than 35 kilograms. And in women, it was a grip strength less than 20 kilograms. And this was also based on predicting poor physical performance. And that was a gait speed of less than 0.8 meters per second. So remember those numbers, 0.8 meters per second for gait speed, and these objective cut points, these absolute cut points for grip strength, because we use those in some applications in some of our analysis and in some other data that we're going to show you in a little bit here. Now, we've been very interested in some of the biological underpinnings of aging and how those may affect some of the changes in mobility, some of the changes in muscle that we see with advancing age. Cellular senescence is one of the hallmarks of biological aging. Virtually all tissues in the body accumulate senescent cells in response to genotoxic, proteotoxic, metabolic, and inflammatory stresses. There are described and measured changes in cell morphology in senescent cells in these different tissues. And one of the hallmarks of senescent cells is that they induce a senescence-associated secretory phenotype, or SASP. This is a combination of pro-inflammatory, anti-regenerative compounds that consist of cytokines, chemokines, matrix metalloproteinase, and other growth factors. So senescent cells occur in tissues. They change their cell shape and express different proteins in the senescent cells, and they also secrete this robust SASP. I've had a long-standing collaboration with Nathan Lebrasser, who's the director of the Kogod Center on Aging at Mayo Clinic. We've been very, very interested in trying to understand how senescent cells influence mobility and aging. And the work I'm going to show you is really a result of that really productive collaboration. We also managed to have a little fun once in a while. So I did convince Nathan back in the winter of 2021 to join me for some really intense skiing in Aspen, Colorado. So this is us having fun. I think we did have some discussions about this work and senescence in muscle and mobility and heterogeneity in response to exercise while we were enjoying ourselves at about 10, 12,000 foot elevation here. So a lot of the work is collaborative work that we've done with Nathan, and we'll hope to continue to do as well. So the first question we asked in collaboration with the Mayo Group is, are the markers of cellular senescence related to measures of physical functioning and muscle strength in older adults? we thought this might be interesting because in preclinical models, if you enriched the SAS through senescent cell transplantation, it compromises muscle performance and physical function. If you pharmacologically suppress the SAS, again, in preclinical models, it's been shown to improve physical function and muscle performance. And there's a number of studies now that have been done that have shown that senescence biomarkers, so markers that are available in the blood, have been associated with chronologic age and clinical manifestations of advanced biological age, such as increased disease burden, adverse surgical outcomes. So Nathan and I took advantage, and this is just to show you this. This was data that the Mayo Group generated, Marissa Schaefer generated, looking at some of the SAS biomarkers in patients that were being rehospitalized for exacerbation of aortic stenosis. So a disease of older age and exacerbation of this disease. And you can see that some of the SAS proteins, IL-6, FAST, TNFR1, GDF15, are elevated in individuals who have heart failure that have aortic stenosis that are being rehospitalized for exacerbations of that condition. And actually, when they did receiver operator characteristics of a panel of 7 of these SAS biomarkers compared to just one of the biomarkers, GDF15, which is a very sort of topical aging biomarker right now. They compared that to a frailty index or just age and sex alone. And you can see the predictive ability, the sensitivity and specificity, the area here is very, very high with the panel of 7 SAS biomarkers, and does much better than either the electronic medical record frailty index or the GDF15 alone here, or just age and sex. So this is a very sensitive and specific predictor of, among aortic stenosis patients, who is more likely to be rehospitalized. So I think this just shows that these biomarkers can be really important in tracking and sort of identifying individuals that are going to have important clinical events in the future. So we took advantage of this panel of biomarkers in what we called the CINO Life Study. We were able, with a grant that we had from the NIA, that Nathan was the PI on, measure a panel of 27 validated senescence-associated phenotype markers. Again, these are comprised of cytokines, chemokines, matrix remodeling proteins, growth factors, and other bioactive compounds. In the life study, the lifestyle interventions for elder study, and I'll tell you a little bit about that in a second, we had samples at baseline, 6, 12, and 24 months. This was a randomized control trial. So not only could we look at baseline associations, but we could look at longitudinal changes and the effects of the intervention. I'll tell you a little bit about that in a second. And again, it was a pretty robust sample. When all was said and done, we were able to get samples from 1,377 participants. This is how they're distributed by sex. And we had other important key physical performance outcomes that were assessed very rigorously as part of the life study, including the short physical performance battery, timed 400-meter walk, isometric hand grip strength, and these were all assessed at various time points as well. And this is just the list of the 27 biomarkers here. The study that we were able to measure these in was the life study, the lifestyle interventions for independence for elders trial. Very briefly, this was a large randomized phase 3 control trial of over 1,600 individuals randomized at 8 clinical centers in the United States. The average length of follow-up was 2.7 years. So depending on when you were randomized into the trial, you were enrolled for just about 2 years to up to 4 years. And the primary outcomes, the primary interventions in this study were really to compare the effects of a multimodal aerobic and resistance exercise training intervention to a successful aging health education control group. And just to tell you a little bit about that trial, the primary outcome was the onset of major mobility disability. And we defined major mobility disability in this trial as the development of the inability to walk 400 meters which meant that at baseline, everybody that was randomized had to be able to complete a 400-meter walk. And then we assessed this every 6 months and documented when a person experienced a failure to complete the 400-meter walk. And this just shows you the main results of the trial, showing that there was about a 20% reduction in the development of major mobility disability in individuals that were randomized to the physical activity or exercise group compared to the health education control group. So we had this data, we had a rigorous set of physical performance measures, and we had the opportunity to measure this really cool panel of senescence biomarkers. Just a couple of other things about this study population, because I think it's a really, it really is a representative older population. The mean age of these participants was 78 years. All were obese or overweight. The average BMI was 30. The prevalence of multiple chronic diseases was relatively high. So again, very representative of a typical older population, maybe not the population you see in many clinical trials of older adults. And they had low physical performance at baseline. So this is the baseline results of the summed short physical performance battery score, right around 7.5. And the habitual gait speed was approximately 0.8 meters per second, so relatively slow as well. Okay, so what did we find with the biomarkers in the LIFE study? So first of all, just as a proof of concept, and this has been done in other studies, we really wanted to see whether these biomarkers were associated with chronological age, even though the age range in the study was relatively low. So activin A, GDF15, and RAGE, and 10 other biomarkers demonstrated significant positive associations with chronological age. And this is largely what we would expect. There were also some sex differences with MDC, PI1, and RANTS being higher in females compared to males. And eotaxin, FAS, GDF15, ICAM1, IL-6, osteopontin, sclerostin, and a couple of the TNF receptors were higher in males compared to females. Nothing too exciting there, but just proof of concept that there were some sex differences and there were some differences with chronologic age. We were really interested in how these biomarkers related to physical functioning, so we relied very heavily on a very well-established measure of physical functioning that was assessed at multiple time points in the LIFE study. This is the SPPB, or the Short Physical Performance Battery. It's a composite measure of physical functioning, consists of a timed 4-meter walk, a timed repeated chair stand, and a very simple balance test. It's very predictive of disability, rates of hospitalization, and mortality. And just to illustrate that, this is the average SPPB score and risk of nursing home admission. And you can see that the risk is very low. If you perform very well on this test, you get a high SPPB score, and that risk goes up as the SPPB score goes down. So what did we find? Okay, so we looked, these are heat maps. We looked at the 27 biomarkers of senescence using Spearman rank correlation coefficients. This is these are the biomarkers related to the total SPPB score, and then we looked at the individual components of the SPPB, the gait speed, the chair rise, and the balance. And we did note that there were several biomarkers of cellular senescence that were elevated in individuals that had lower SPPB scores. And you can see that here with the strongest association being for activin A. And you can see the graph here looking at total SPPB score at the top and activin A here. MMP7 was the most highly correlated with slow gait speed. ICAM, which is a calcium adhesion molecule, was positively correlated with better chair rise time. So this is a bit of an anomaly here, but this is what we found. You can see that up on the heat map here. And again, higher levels of activin A were related to lower levels of balance. But you can see overall, there were multiple senescence biomarkers that were related to low physical performance on the SPPB on the SPPB test. And we also looked at the gait speed during the 400-meter walk test because we had that data. And we saw a very similar pattern with the 400-meter walk as well. We did gradient boost modeling to look at the relative variable importance and which of the biomarkers had the most variable importance in predicting the total SPPB score or the individual components of that. And you can see that many of the senescence-related biomarkers were more predictive than some of the well-known predictors of low physical functioning, poor physical performance, such as sex, chronologic age, and BMI here. So we see that for the overall SPPB score, many of these do better in the gradient boost modeling analyses. And the same is true when we look at the individual components of the SPPB score here. The chair rise on the right, gait speed as well. BMI is a little bit stronger predictor here, but again, still some of the biomarkers here have high variable importance here. And the same is true for the balance measures as well. We went on to generate receiver operator characteristics using these biomarkers. And what we did is we asked the question about how well these biomarkers would predict an SPPB score of 7 or less. Okay, so that's a pretty low level of physical functioning. And if you look here on the left, this is the training model, and on the right is the validation model. And the curves here in dark blue, this is model 1 that had the top 10 biomarkers based on the gradient boost modeling, plus age, sex, race, and BMI. So everything here, you can see that we get an area under the curve of about 0.93 for predicting low SPPB score. If we just look at the top 10 biomarkers alone, they do just about as well. Area under the curve about 0.92, and they do clearly better than when we look at some of the known predictors of poor SPPB scores, such as age, sex, race, and BMI. And remember, this is the line of identity. This would be equivalent to a flip of a coin. So the biomarkers they're random. And we saw that this pattern hold up in our validation set. This was a subset of participants from life that we looked at the validation here. Again, the top 10 biomarkers, either alone with the known covariates, did pretty well, much better than just the known covariates of poor physical functioning here, and much better than a random flip of a coin. We did look exclusively at a slow gait speed, again, a gait speed below 0.8 meters per second. Remember that from the Sarcopenia Definitions Outcome Consortium. And in fact, just gait speed alone, the biomarkers didn't do as well predicting slow gait speed as that did for the total composite SPPB score. You can see here again, this is for the biomarkers plus the covariates. This is for the biomarkers alone, and this is for the known covariates: age, sex, race, and BMI. And this really kind of fell apart in the validation cohort as well. So not as strong a predictor for gait speed individually. We also wanted to look at muscle weakness. So we looked at isometric hand grip strength here. And again, we saw a number of senescence-related biomarkers that were associated with low grip strength in both females and males. And the most significant predictor in females was the biomarker RAGE, and the most significant predictor in males was again sclerostin here, again with a positive association, so a little bit contrary. But again, we did see a number of the same biomarkers we saw that predicted low physical performance, predicting muscle weakness by grip strength here, such as activin A, GDF15, MMP2, some of the TNF receptors, and interleukin-6. Again, we did gradient boost modeling to look at the variable importance here, and looked at and came up with the top 10 biomarkers of variable importance with the GBM modeling in both females and males. Interestingly, we did get a different pattern of variable importance with the grip strength. So it looks like different sets of these senescence biomarkers seem to be influencing muscle strength compared to when we just look at the measures of physical performance. And we're still trying to understand this, and hopefully in our future studies, we'll gain some more insights about this. Again, we did some ROC characteristics here for poor grip strength in females and males. And again, these are the cut points that came out of the sarcopenia definitions and outcomes consortium of less than 20 kilograms in females and less than 35 kilograms in males. And again, the models generated areas under the curves of about 0.74, 0.72, depending on whether you used all the biomarkers with the covariates or the biomarkers alone. These were, I think, nominally better than just the known covariates here with the, predictions of about 0.66 as well. And then this was true both in males and females. So from this initial analysis, I think we were really excited about this results that, the strong associations between biomarkers of the senescence of the SASP, which is a biologic hallmark of advancing age and measures of physical function and muscle strength in a well-characterized cohort of older adults. These findings may offer some clinical utility in identifying older persons at risk for mobility disability and poor physical functioning. And we may also be able to leverage some of these data in clinical research to identify persons who may be most responsive to function promoting interventions and could potentially also ultimately serve as surrogate endpoints in clinical trials that are designed to improve and enhance late life health and function. Obviously, there's some limitations in these analyses, including the fact that this was a cross-sectional design. So it is a challenge assessing causality. There are numerous multiple chronic conditions that these individuals have. It was a pretty finite age range of study participants. We excluded individuals in the life trial that had higher levels of physical function and better health status. So it may be difficult to dissociate what we're seeing from the effects of disease burden. And, again, as always in clinical trials, this is a self-selected population and may represent a unique population of older adults. So following up on that, we did some additional analyses. We really wanted to look at the ability of some of these biomarkers to predict some of the primary outcomes in the life study. The primary outcome that we looked at was the onset of major mobility disability. I'll show those data. And then also really ask questions about whether the active intervention, the physical activity intervention, had any influence on some of these biomarkers over time in the two randomized groups that we looked at. And then ultimately, as we're moving into the new project, we thought that this could be useful to assess non-responders and responders to the intervention, in particular to physical activity or exercise interventions. And I'll get to that a little bit later on. And we were able to show that our biomarkers were able to predict longitudinally the onset of major mobility disability. We looked at incident and persistent mobility disability. Incident mobility disability was really the first occurrence of the 400-meter walk failure. Persistent mobility disability was when an individual had two subsequent failures in the 400-meter walk, or a 400-meter walk failure and then death after that. And in both cases, we saw a similar panel of important biomarkers using the gradient boost modeling. And again, some of these performed even better than some of the known predictors of onset of mobility disability, like chronologic age and education and race. This was for, at the top here, at the top is for incident mobility disability, and these are the data on the bottom panels for persistent major mobility disability. And then we assigned the biomarker concentrations to quartiles and compared them to the lowest biomarker quartile 1. And you could see that there was a significant increased relative risk of onset of incident and persistent mobility disability in individuals either in the second or third quartile of biomarker concentration here, depending on the given biomarker. You can see for activin A, VEGF, TNFR1, MMP7, GDF15, IL-6, and ICAM. Again, inverse association here for ICAM. So that's consistent for what we saw with the physical performance measures cross-sectionally. And again, the same is true for persistent mobility disability here, predictive in the highest quartiles of the senescence biomarker concentrations here. So we've got a nice set of robust biomarkers that predict cross-sectionally and longitudinally some of these bad physical performance outcomes. What about the effects of exercise and the intervention? The Mayo group had done a small study, non-randomized study, looked at some of these biomarkers in a group of fairly healthy older adults that were engaged in a lifestyle exercise intervention. This is work that Davis England did in the Labrasser lab, showing that a number of these senescence biomarkers were actually significantly reduced in response to the exercise intervention. And with some latent variable analysis, we're able to show that the biomarkers could predict responders and non-responders. This was a pretty healthy cohort. And this was also not a randomized study. So everybody got exercise in this group. So I think it's some interesting preliminary data, but I think some more work really needs to be done to understand these markers of cellular senescence and how they respond to physical activity. In the LIFE study, when we looked at changes in the biomarkers in individuals that were randomized to the physical activity group versus the health education group, we really didn't see any significant changes in the biomarkers in response to people being assigned to exercise or the physical activity group. There was no change. But we also did was we had data on accelerometry from virtually all of these participants. So we knew how much voluntary physical activity they were participating in every 6 months during the intervention. So we looked at quartiles of moderate to vigorous physical activity from direct accelerometry measurements in individuals that were randomized both to health education or control here. So these are the quartiles that we set up from low physical activity to higher, moderate to vigorous intensity physical activity. So we compressed the randomization. But if you look here, you can see that in general, the individuals in the higher quartiles of physical activity participation, there are more individuals from the physical activity group than there are from the health education group. And the converse is true for the low physical activity group. Okay, so, and what we were able to show was that in the individuals that were participating in more physical activity at 12 months, they had significant declines in several of the biomarkers of cellular senescence. And those are listed here. So these are compared to quartile 1. So this is quartile 2, quartile 3, and quartile 4. So in many cases, the higher quartiles of physical activity participation resulted in greater reductions in the senescence biomarker concentrations. This was true at 12 months, and it also persisted in individuals at 24 months. So no direct effects of randomization for exercise, but at least a signal that more physical activity participation was associated with lower senescence biomarker concentrations in this mobility-limited, older cohort of men and women. Nathan's group, and we don't know what the biological tissues are that are producing these senescence biomarkers. They can be coming from adipocytes, fibroblasts, muscle, other tissues. We really don't know when we're measuring them in the plasma. Nathan's group has done a number of really nice studies showing that there are increases in markers of cellular senescence in skeletal muscle in very specific subpopulations in skeletal muscle. These are just some data from humans from a paper they published in Nature Aging in 2022, showing that TAF is upregulated in older skeletal muscle. P16, a marker of cellular senescence, is upregulated in older skeletal muscle, and P21 expression as well here. What's interesting is that there's only specific subtypes in the muscle that are expressing markers of cellular senescence. And I think going forward, this is really going to be where it's going to require us to look at individual nuclei from muscle biopsies that we take and be able to identify which cells are expressing senescence proteins, as if we want to go on and do more studies in humans and really try to understand also how that muscle is responding to interventions like exercise. They were also able to show that expression of one marker of cellular senescence, P21, was actually inversely related to leg press strength in these individuals. So higher P21 expression was related to low leg press strength in these individuals. So what about exercise and more specific types of exercise? And our laboratory for a number of years has been interested in resistance exercise training and exercise training overall. But what is strength training and what is resistance exercise training? And the first thing to remember is that skeletal muscle responds and adapts to loading conditions. High force, high power output types of activities induce muscle hypertrophy and gains in muscle strength. Low force, inactivity, bed rest, and zero gravity induce muscle atrophy. And this is very important because we know that with aging, there's a loss in muscle mass and a loss in muscle strength. And embedded in all the guidelines for exercise and physical activity for older individuals, both nationally and internationally, is a recommendation that older individuals participate in strength or resistance training. In our hands, what this means, and when we do this in the laboratory in a very controlled situation, we really prescribe and supervise the exercise. So individuals are exercising 2 to 3 times per week. They engage large, major muscle groups of the upper and lower extremity. The magic formula forever and ever has been for individuals to perform 3 sets of 8 to 12 repetitions, so that by that 10th or 12th repetition, people are reaching their point of fatigue. So this is typically a relatively high intensity exercise intervention. And we believe, and others have said, that to maximize strength gains and hypertrophy, the training does need to be performed at a relatively high intensity, 70 to 90% of the 1 repetition maximum muscle strength. And again, this is approximately the number of contractions needed to reach fatigue with about 8 to 10 contractions. That's not the only type of things that you can consider to be resistance training. So if you're in the community or you're going to your local gym or YMCA, all these other things on the list count as well. You can get these effects using exercise bands, weight machines, handheld weights, doing calisthenics, digging, lifting, or carrying things like gardening, and even some forms of yoga and Tai Chi count. So I don't want people to come away thinking this is the only type of resistance or strength training exercise that you can do. But this is the kind that we do, and we're trying to understand and evaluate this in a very carefully controlled way in our laboratory. So what are we going to do going forward? And this is where it gets to this personalized approach. We've had this data for a long time. This is a study where we gave older adults that had mobility limitations 6 months of resistance exercise training, and half the group got a placebo intervention, half the group got whey protein supplement. And that's not important for this discussion and for this seminar, except that the whey protein really didn't have any effects on the strength or mass gains that occurred with training. These are the data on the changes in muscle cross-sectional area from the CT scans that we performed in individuals at baseline and at 6 months. So after the 6 months of the exercise intervention. These are the 2 groups, placebo and whey. Whey did a little bit better, but it wasn't statistically significant. And there was a statistically significant improvement in muscle cross-sectional area, either total muscle, or if we separate it out by differences in optical density, these all improved. Normal density muscle went up, low density muscle tended to go down a little bit or didn't really change. But what's really interesting is I also plotted the individual changes here in both groups, with whey and with placebo. And I've always, ever since we generated this data, which is now over 10 years ago, I was always struck by how much variability there was in this response. And, we know that, one of the biggest influences of how people will respond and adapt to exercise interventions really is whether they'll do the exercise or not. So whether they adhere to the exercise or not. So yeah, some of this variability could be because of that, but we went on and we did analyze the data in a little bit more careful way. And what we did is we said, okay, let's take out all the people that really didn't come to more than 80% of the exercise sessions. So those are all the poor adherers to exercise. So let's take them out and let's look at the variability or the heterogeneity in response a little bit more carefully. Okay, so this is with 6 months of resistance training. And I was really sort of surprised by this data. So these are the individual subjects in this study when we excluded individuals that really adhered poorly to the exercise. So these were all the completers, if you will. And you can see here, the top panels are the change in muscle cross-sectional area here in men and women. And those changes range from a 10% loss to about a 17% gain in muscle cross-sectional area or size. These are the changes in leg press strength in these individuals, which ranged again from about a 14% loss to nearly a 94% increase. And again, we also examined the changes in physical functioning using the short physical performance battery score. And again, that variability range or heterogeneity range from an 11% loss to 100% increase. So all these people were doing a lot of resistance training, yet some people lost mass, didn't improve very much, or improved a whole lot. So what's going on here? I think this is really one of the questions that we're really interested in understanding. We also did another study where we did resistance training, but we also had an active control group in that group. We were able to show that strength, power, and muscle fatigue was improved in the group that got resistance training. This was for 12 weeks compared to a group that got just a flexibility home-based stretching program in this study. And when we again looked at the variability or heterogeneity in the response in those individuals that were very adherent to the program, in the individuals that got progressive resistance training versus the flexibility training group, again, in the group that got exercise or resistance exercise here, there was a 23% loss to a 33% gain. And this was a little bit attenuated in the flexibility group, but also important to note that the control group also has its own inherent heterogeneity to response. And I think going forward, as we're trying to study that, we also have to include an appropriate control group in our study design to really evaluate what the real variables are that contribute to this heterogeneity in response. So we had this really nice data on senescence and physical functioning. Nathan's group's been able to show that senescent cells exist in muscle and in human muscle. And we know that individuals have varying heterogeneic responses to resistance training, which is an important component of the exercise prescription for older adults. Well, good things happen once in a while. The NIA also thought that this was an important area of research. So they had convened a workshop in 2021, which really was focusing on understanding the heterogeneity of response to exercise and approaches to optimizing the clinical efficacy of exercise training in older adults. I didn't go to this workshop, Nathan did, but this was the paper that came out of that was published in 2022. Usually, in their wisdom, what the NIH and the NIA do is when they have one of these workshops and they publish a paper, usually they can effectively issue a request for applications to fund proposals in this area because there's interest, the community is interested, and they think this is an important area of future investigation. So a request for applications was issued in August of 2023. The RFA was entitled Elucidating Variability of Physiologic and Functional Responses to Exercise Training in Older Adults. The RFA was issued in August of 2023 with a due date of November 6th, 2023. Now, importantly, Nathan and I have been talking about the data we had on senescence, biomarkers of senescence, and this data that we had been working on with some people in my group, Marissa and others, on heterogeneity, variability in response to resistance training. So we at least had the ideas in our head to try to put this application together. Again, our grants office here at Tufts, Megan Faraday and her group had alerted us to this RFA. So that was really, really important because, again, you can see the timeline here is always very tight. So we submitted the grant and surprise, surprise, we just got it, which is great. This is our grant. This is the title of the grant. It's called the BRIO trial for short. We just got the award recently. Nathan and I are MPIs on this grant. We have a really good, important team of investigators that are working on this, including Ian Lanza, who's a metabolic expert at the Mayo Clinic, Tamir Chandra, who's an expert in DNA methylation and DNA clocks, Stacy Witham, Beth Atkinson, and Amanda Tweed. And at Tufts, we have a great team with Sai and her expertise in precision nutrition and metabolic regulation and energy expenditure. Lisa Seglia with her work on hormonal responses in aging and intervention trials, and Marissa Peasley as part of my group as well. And I just wanted to share with you the aims of the study, because I think this is going to be really, really interesting and really builds on some of the work that I've shown you. We really want to critically examine biological, clinical, and behavioral mediators of progressive resistance training associated exercise response heterogeneity in physical function in older adults. We want to precisely define the variability and changes in physical function as measured, again, by the short physical performance battery. The RFA was very specific that we had to have a clinically meaningful primary outcome, and we also had to have a physiologic or biologically meaningful primary outcome as well as part of the trial design. So we decided on our primary biological outcome, clinical outcome being the short physical performance battery, but then we also wanted to measure biological outcomes that may predict this change in short physical performance battery score, including measures of cellular senescence and DNA methylation, clinical variables, as you see listed here, some behavioral factors in individuals that are going to be randomized to the progressive resistance training intervention or the health education control group. This is going to be a 6-month-long clinical trial, assessments at baseline, and 6 months. Individuals will get supervised resistance training or a health education control group during that prolonged period. It's going to be conducted at both sites, both at Mayo Clinic and here at the USDA HNRCA at Tufts. We're going to apply a very interesting personalized precision approach path analysis to examine responders and non-responders. We are also going to, as part of the outcomes in this study, look at metabolic variability by looking at the variability in changes in insulin sensitivity derived from the glucose and insulin responses to a mixed meal tolerance test, again, comparing baseline and 6 months in individuals that are randomized to progressive resistance training and health education. And we'll look at some other secondary metabolic outcomes as well, using the same precision approach to in our statistical analysis to look at responders and non-responders to these variables. We do also have a number of really cool secondary outcomes in the physical function space and the muscle space. We're going to look at the heterogeneity again in response to muscle size using, again, computed tomography, CT scans to look at muscle cross-sectional area and muscle density. We'll measure muscle strength and explosive muscle power in these individuals and look at 6-minute walk distance, as well as a number of other physical performance measures and isometric grip strength. So we're really excited about this study. We have no data. We haven't really started the clinical trial yet, but I just wanted to share with you this exciting work and try to show you how it really built on some of the preliminary work that we had done using the life study cohort and some other data that we had from our own clinical trials here at the HNRCA in my own lab. Just a number of grants that are supporting us, including the new grant now, and I'm happy to take any questions with the rest of the time. Thank you.
Thank you, Roger, for such a wonderful presentation, and congratulations to you and Dr. Bresser for your recent NIH R01 award. I think this will be a really exciting study. So with that, we already have questions coming in. We are close to the end of our time here, so we'll just run through those questions quickly, if you don't mind.
Absolutely.
Okay, so I'm going to start with Christopher Wiley. He says, great talk, Roger. One concept I have struggled with in the senescence field is that while we see subsets of SAS biomarkers being enriched for several degenerative diseases, we rarely see the same systemic factors as being the most associated with degenerative diseases. On one hand, this could be a function of the inherent variability of the senescent cells, but the other possibility is there are processes that drive these markers that happen to also associate with subsets of the SAS. Are you able to differentiate between these possibilities in your study?
Yeah, not with the biomarkers. That's a great question, Chris. And obviously, we don't, with the biomarkers that we're measuring in the plasma, we don't know where they're coming from, and why some of those are associated with some of the bad outcomes that we measured related to function and why others are not. I think it's reassuring, at least to me, that there are a cluster of SAS proteins, at least in our studies, that did predict, weakness, poor walking speed, and poor balance. So that's a little reassuring. We are going to do in the new study, we are going to take muscle biopsies from all of our participants, and we're going to do single nuclei RNA-seq on them. So we will be able to look at least on expression of features of the SASP and DNA methylation in specific cell types. When you take the biopsy, there's multiple different cell types, even within what you're calling muscle. So I think that will be a really important tool to help us dissociate that, at least in the muscle space, but not for other tissues for this study.
Great, thank you. Jose Ordovas says, thanks, Roger. Excellent talk and excellent work on measured biomarkers. He's got, Jose, in the interest of time, we're only going to ask one. What is the current understanding of genetic risk factors for sarcopenia, and how can this information be used to develop personalized prevention strategies through diet and specific physical activities? Yeah,
great question, Jose. And there are a number of studies looking at risk factors, genetic risk factors for sarcopenia. There are some SNPs that appear to be significant, and also SNPs that are also predictive of response to function promoting interventions. So we are not necessarily focusing on that in our study because we had to pick something, but there is data suggesting that there are genetic risk factors for sarcopenia, and probably also some early life effectors like unique DNA methylation, post-translational factors like DNA methylation. We know that grip strength early in life predicts who gets weakness later in life, similar to bone density. So yes.
Great. We have a question from Perry Ecker. Thank you for the great talk. Have you ever tested the combination of exercise with an inhibitor targeting your top SAS biomarkers? Is it possible that some of these biomarkers are mechanistically involved in exercise's effect on senescence and muscle health span, and their blockade activation could impact the benefits of exercise?
That's an excellent question, Perry. Thank you. We have not. But I will tell you that we are very interested in whether function promoting therapies, pro-myogenic therapies, whether they do behave synergistically with pharmacologic interventions, whether these are pro-myogenic interventions or senolytics or other types of therapies that might affect the SAS, senescent cells or components of the SAS. I think there's more to follow on that, and there's a lot to learn about in preclinical studies before we proceed to clinical trials, but we are very interested in those kinds of synergies. I think that's going to be what people are going to look at in the future.
Great. And we have one more question from Larissa Constantinou. You mentioned RAG as a biomarker. I am curious whether exogenous dietary AGs may affect RAG concentration in the body, aging, and performance.
That's a very good question, and I do not know the answer to that. Thank you.
Okay, and we have one final question. This is the big one. From Michael Mong, who says, in other words, what are your thoughts as to what might be causing the muscle loss?
Muscle loss with aging is probably caused by, just like every complex syndrome of older age, muscle loss with aging is probably caused by some underlying biological features that are expressed with aging, and a number of profound environmental influences, like the increase in sedentary time that occurs as people get older, the decline in spontaneous and voluntary physical activity, changes in dietary intake, the reductions in dietary protein intake that occur at least in some older individuals, and probably some of the increased burden of chronic disease that all have sort of pro-inflammatory effects that could also be affecting muscle atrophy and muscle weakness. So I think it's multiple things, and that gets hard to sort of tease out in human studies, as I'm sure you're aware.
So one last comment, then we'll close this up. From Richard Alpert, no question, just thank you for a fascinating talk and congratulations on your grant. And I think that's just a wonderful way to end today's seminar. Thank you, Roger, for a stimulating talk. And again, congratulations on your recent GSA award and your grant. Thank you very much, everybody.
Thanks for everybody attending.